Thank you. So I changed the title a little bit. Anyways, so I am supposed to introduce myself. I'm Hae Kyung. I'm at the University of Chicago in the section of genetic medicine. And here is my overarching goal. So my advisor told me I need to have a vision, but I don't write vision because it sounds like a business school type. So anyways, so my goal is to develop methods to make sense of these huge amounts of genomic data and other sources of high-dimensional data with the ultimate goal of making discoveries that can be used to improve the health of people. So that's kind of the guiding principle of my research. Here is my team. So except for Alvaro, who had to stay home taking care of his baby, or the other ones you should be able to see, and if you don't see them, let me know. So Rodrigo is here, Jamal, and Milton. Okay. So, okay, let me start with prediction of complex traits, because I thought that for this crowd, like at least part of this crowd, this would be more interesting, or at least to start with. I have 3 parts. Let's see for which parts I can go through. And please interrupt me, ask me questions. I was told that I had been asking too many questions, so, you have your revenge. No. So I thought this was a nice way to kind of get some context. So in year 2000, President Clinton announced the completion of the first draft of the human genome. And he said that it would revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. And Francis Collins followed up by saying that diagnosis of genetic diseases would be accomplished by 2010, and that treatments would start to roll out he added perhaps, in 2015. So why are we not there yet? So, because prediction is hard, especially of the future, in any case. So, well, to be fair, so let's go to about the complex trait architecture, complex genetic. I think that title is wrong. Anyways, let's continue. So back then, what we were expecting that about the genetic architecture of complex trait is that we would find these large effect sizes, genes that with very large effect sizes, and then once we found them, we would be able to target them and try to develop treatments and drugs for the diseases. But instead of that, beautiful skyline of this is the city of Chicago, what we found is that the architecture is like this, not very interesting for most of them. And now we have Jonathan Pritchard saying that it's actually all the genes are causal. So if all the genes are causal, who cares, right? Is this any use? Can we make any use of this? This is kind of, okay, let's see whether this will work better. Because you are getting the next slide's title before I get there. So I like showing this picture. Let's see, John is not here. I check. I like to show this figure as a way to show that, even if you don't know what's the effect size of each one, like the accumulation, if you are smart enough to accumulate things just the right way, you may be able to find this beautiful or hidden structure in the data, and that's the goal, like the kinds of things we would like to do. No, this is not working like that. Okay, so into whole genome prediction models. So this is what we all assume, an additive genetic model. And probably people who are doing deep learning find this not satisfying, but there are many reasons why we want to stay in the linear realm, but I would like to hear from other people ways to improve upon this. But this is what we do for now. And then, so Y here is the phenotype. X is a genetic marker, let's say SNPs, to simplify things. And we sum over, 1 million SNPs or 10 million SNPs. And then these betas are the effect sizes. So, and then this epsilon is the, the remaining that we sweep under the rug. And so, well, some people call it environment and everything that didn't fit in this additive model, of course. So if we want to do prediction and someone told us which ones are these betas, then we are done. That's our prediction. So many of these prediction models will be ways in which we're going to estimate these betas. So the simplest thing you can do, take Y, regress on each one of the SNPs, take the effect size, and use those as your beta as if they were the truth. And so what you are doing in that case is least squares. So you can be a little bit more sophisticated, do penalized regression, use all the SNPs at the same time, and there are a bunch of methods that do that. Let me go through that. So for example, the very simple, what people call polygenic trait ways. So this was one of the first examples that I could find, at least, where they use what people call polygenic risk score. Essentially, you run GWAS, from your Plink output, you take these effect sizes, multiply by the SNP dosage, and that's your prediction. So they did that back in 2009, the International Schizophrenia Consortium, and they used this. So what they did is they ran GWAS, they plotted their Manhattan plot, they found absolutely nothing, it was very flat, very disappointing. But then what they did is they took one set to run the GWAS, an independent set to test, and they found that this risk score was predictive of schizophrenia and bipolar risk. So then they concluded that these traits are polygenic. So this very simple approach does give you some predictive power. So this is an example of the polygenic risk score. And if this is something you already know, let me know and I can go faster. No complaints. Okay, so penalized regression. One way is ridge regression, where you do least squares. So this is the least squares of Y minus your predicted version, the L2 norm. And then you penalize by adding so you don't want the sum of squares of the effect sizes to be very large. So that gives you ridge regression, which is equivalent to running the mixed effects model, calculating best linear bias predictor. But anyways, let's not go too much into that. And then another approach is to use lasso approach, where what you penalize, you minimize your error squared, but you want the L1 norm. So you want the sum of the absolute values of effect sizes to be small. And elastic net it's a mixture of L1 norm and L2 norm penalization. So those are several variants, and then once you have your betas, you plug them in, and there is your prediction. A variant of this, you can go Bayesian, and this is a paper by Matthew Stevens, where they split this into two terms. One term is a bunch of sparse effects, so a few SNPs that have large effects, and then the other term is the polygenic background. So this is a nice split of the data, and it gives you nice things like sparsity of the genetic architecture of this trait. So in others, let me just continue. And this is something we propose to include, in addition to the genetic component, transcriptomic, and other omic components. You can just add them up, represent it in similar fashion, and you can fit this model using restricted likelihood. Anyways, so because these traits are so polygenic and the effect sizes tend to be small, back in 2013, people calculated so that we could do prediction, because we know that these are heritable traits, so we can predict this. But so the upper limit of the prediction is the heritability, right? The SNP heritability, which is the heritability that is explained by the SNPs that is in your genotyping array. So, so if we want to reach that maximum, so they calculated what kind of sample sizes we need, and the conclusion was we need millions of samples. now we are in 2018, and this paper that we discussed in the Journal Club, we are at that stage for height that's highly heritable. We are hitting that upper limit of heritability. Here, these authors, they use just simple lasso, fitted the model in UK Biobank, where you have 500,000 people. It's not millions, but 500,000, so we are reaching that limit. Another paper that just came, a preprint, where they predict coronary artery disease risk using 1.7 million markers. And they show that by using this you can use as a biomarker to predict risk of cardiovascular disease. So the top 20 percentile of the people have fourfold increased risk of having coronary artery disease relative to the bottom 20%. So this is a lot more predictive than LDL cholesterol, so your bad cholesterol level, for example. So this has clinical implications. Another type 2 diabetes, like very, very polygenic trait that has been very hard to find anything for the longest time. And now just the polygenic score, and this is just a simple polygenic score. You run GWAS, you take your betas, you do some thinning, so to avoid using multiple SNPs that are highly correlated, and that's your predictor. Again, this one.
In the previous paper, how many SNPs do they use?
To compute, they use 1.7 million. So they use, they have started with 4 million or more. I don't remember, but they end up picking this 1.7 million.
And that's based on?
They do GWAS results, GWAS results, directly the GWAS, univariate regression. And what changes is what threshold you use. So they don't use all p-values. They do some thresholding, only the significant ones. And then how you do the thinning, so that you don't get too many that are highly correlated.
The sample sizes are coming from the UK Biobank. Is that what they're using?
They use UK Biobank as a test set, and they use an independent cardiogram. They combine several.
So the UK Biobank as a test or as a training program?
As a test, not the training set.
Oh,
as the test, because
So they had a bigger set than UK Biobank?
So the large meta-analysis of coronary artery disease, because UK Biobank is this large biobank with 500,000 people, but the cases are not as many as what we can achieve in a case control study. So, although coronary artery disease is pretty prevalent. So simple polygenic score, who would have thought that you get pretty good prediction. So this is the first part. So complex traits are highly polygenic. Effect sizes are very small. We need very large sample sizes. That's the conclusion, take-home message. So okay, next we apply,
There was a paper put out not too long ago that questions the notion of using polygenic risk scores. At least across ethnicities, were these all in European samples?
They use Europeans, because across populations it would get worse, So predicting, so we are going to use, we are going to talk about how to predict gene expression traits, and then in the third part I will show you what we use that for. So for gene expression traits, because we have a location in the genome, so we can split the local versus the distant. So the genomic region that's nearby the transcription start site of the genes, and then the piece that's farther apart. So that's what we are going to see to do. And then in order to do prediction, we want to know a little bit about the genetic architecture, because depending on the genetic architecture, different models will work better for prediction. So as I was saying, we split the model, the additive model, into a local component nearby. So these are all the SNPs that are 1 megabase away from the transcription start site, for example, and these are the remaining SNPs. So first thing we do is we calculate the heritability, the local versus the distant heritability. So what we do is we calculate the covariance. So we use mixed effects models where we assume these betas are random. So normally distributed, we calculate the covariance of the SNPs in the local area, in the local genomic region. We calculate the covariance in the farther apart, and then we use restricted maximum likelihood to estimate the variance corresponding to this, the local component, the variance corresponding to the distant component. That way we'll have heritability, local heritability, and distant heritability. And we use GCTA method. We have the author here. He lifted his head when I said that. Anyways, we use it as a calculator, which is good. So the data set we're going to use is from one that Alexis that gave the talk yesterday described. No, she didn't describe that, but she published on this. It's 922 individuals, whole genome, whole blood RNA-seq data. Yeah. Okay, so this is the estimated local heritability. Here, this is ordered by estimated heritability, local heritability. That's why this curve is just increasing. And the gray bars are confidence intervals, 95% confidence intervals. So we can see that for some genes, the heritability is not different from 0, but for these genes from here on, they have heritabilities that's greater than 0, some close to 1. So there's a range of heritability values for different genes for the local region. For the distant one, what can we say about this? There are a few here where there's not, seems to be. So we kind of drop that part for now. Until we have bigger sample sizes or better methods, we don't deal with that. We just focus on the local component.
You could treat every gene as independent observation and just average those things out.
Yes, you can do that, actually. Yes, that's what Arcus did. You can get an average heritability across genes, but it doesn't tell you much about each gene. So you can tell whether so that's what Arcus used to estimate what's the contribution of the distal versus local component. But that's averaged across genes, right? Yeah, so there is something there, and people talk about 70% of the variability in gene expression being due to trans, but we are only looking at local for now. Yeah. So another thing we wanted to do is to study the sparsity or polygenicity. Is it just a few SNPs that are affecting the expression of a gene, or is it all of them in aggregate that's doing that? So the way we thought about it, so the lasso, elastic net, and ridge regression, we can think of elastic net as kind of comprising also lasso and ridge as extremes of those, right? So elastic net is the mixture of these two, L1 and L2 penalty. Lasso is L1 penalty, ridge is L2 penalty. So depending on what the proportion of L1 and L2 are, we thought that would give us some sense of the sparsity or polygenicity of the trait. So this is graphic representation, cartoon of that. So if on the left, this is only L2 penalty, lasso is L1 penalty, and in the middle somewhere elastic net, depending on the mixing parameter. So we thought that if we plotted the performance of the prediction models, we would find that if some genes that are highly polygenic, we would find that ridge regression would do better than lasso. If it was the other way, then if it was very sparse, then lasso would do better than ridge. And then if it was somewhere in between, we would get some kind of maximum. So that was our, yeah.
Would that be another dimension to look at the magnitude of the
Yes,
L1
yes,
and L2, right?
yes, it's true. But yeah, let me get to, This is just one way to look at this, and I agree that you need more than that, yes. So we ran this for all the genes. Here is some 340 genes in chromosome 22, and this is what we found. The prediction performance was pretty much flat for every gene. So each curve is a gene, and then you can only see this a little bit of dip for for ridge regression. So we conclude from here that it's probably more sparse than polygenic.
So I was also wondering if you had like epigenomic data in this cell type, can you split your your lambdas into two pieces where SNPs that are inside elements get less penalty and SNPs that are outside elements get a much stronger penalty. So
Yeah.
you might not get better performance, but you might get like more interpretable models.
Yes. So yes, there are many ways you can go about that, but yes, definitely it's something that we are looking into.
One more question about that figure, the previous one. The sample size kind of influences the results.
Yes,
So
yes,
large sample size, probably all of them will be the same. So in other words, like
it may be, yeah. It's like mixed effects models being robust to capture
I was
that.
thinking more like if you have lots of data, lots of individuals, then any
it doesn't
model would
matter.
actually
We'll
figure-
do
yeah.
well. Yeah,
So
you're
maybe,
probably right.
The difference between these models might be better with a smaller sample size or some
That's
some
true.
way you can discriminate
Yes.
results by
Yes, that's true. But given limited sample size, we want to use the best model. Yes.
So is the x-axis here?
The mixing parameter, it's between 0 and 1. If it's 0, it's ridge regression. If it's 1, it's...
So you have the same, the sum of the L1 and L2 is constant, and you're just changing the mixing?
Yes,
But,
yes.
But it seems like, and going back to my previous question,
Yeah.
it seems like the open question is if you varied that sum,
Uh-huh.
how much regularization you're doing.
Yes.
You might see very different effects in different parts of the space.
Yes. So this is...
It looks like there's hardly any effect at all, one way or the other.
Yeah.
It seems like maybe there's not enough regularization happening.
Yes. So we find that lambda by cross-validation, like using GLMNet, but yes.
Oh, I see. So it's a different.
Yeah, this is just to study the kind of, we fix the, that's, it's, we fix the mixing parameter, and then we let the lambda, the total lambda vary, and the one that maximizes, yes, okay, yeah, that, we do that, yes. Yes.
Do you test for things like peer factors or SBA?
Yes, we already, yes, we do peer factor, the same thing like GTEx does.
Do you try to predict after you've adjusted out the peer factor, or are you testing on things that are unadjusted for the peer factor?
So when we test, this may be after, but I have some plots where we do test against unadjusted ones. Yes, but yeah, we didn't do this study. Yeah.
Just to better understand this, so this will be the mixing between the proportions?
Yes, it's a mixing alpha. It's between 0 and 1. 0 means.
Okay, and how it will compare to, I mean, also as a limit case, if you were only to pick one for each one?
How we pick one? So
No, if you were to pick just one SNP per gene, like
if I yeah. So we did that, but not in this context. But yeah, we compare the prediction models just using one SNP too. I mean, the idea of this was we would find all these nice plots, but then we found much boring results. But anyways, but still, this was told us that ElasticNet would be good, Lasso would be good probably, but we picked ElasticNet 0.5 because that gives us a few more SNPs, and if there's missingness and not good quality SNPs, then that would our hope that would help us kind of be more robust. Yes.
Is this imputed SNP data? Do you have enough SNPs per gene to really achieve some?
This is, I mean, I don't think it makes much difference. I can't remember whether this, this was imputed, yes, imputed, yeah. Okay, so this is the prediction R-squares calculating the observed, so correlation between observed and predicted squared, and then ordered by the estimated heritability. So the black thing that you can't see is the heritability estimates. They just continue to grow because we order them that way. And so the red dots are the correlation square. So maximum is 1, 0 means we don't predict at all. 1 means we have perfect prediction, at least in the correlation sense. And we can see that the correlation, the performance tracks the heritability, which is expected.
What data did you use?
Sorry?
What data did you use?
So DGN, so it's depression data where we have 922 RNA-seq data.
So that's the average across all those points. But if I were to, let's say, pick a threshold of prediction accuracy, let's say 50%, how many of the genes pass that?
So you can, well, it's not ordered, but if I put here, so it would be those dots there. 50% is, I don't know, but it's not that many. 1,000, less than 1,000, I'm guessing. Yeah. Like I think half of the genes were at 1% R squares. And then here is some examples of predicted expression. So y-axis is predicted, is observed. X-axis is predicted. And these genes are picked, in this region, because I don't want to show you just something that looks like noise.
And is it the case that the top left one is just one SNP?
That's what's going on. It's not one SNP. We are using, actually, yeah. So here we are using HapMap SNPs. We want it to be restricted to that SNPs for application later. And what's going on is that HapMap doesn't have that SNP, the causal SNP. But this, what it's doing is it's predicting that causal SNP, and that's giving us this thing. That's,
but the ground truth would be there that there's a single...
There's a single SNP that is under balancing selection or something. There was a paper on that, actually.
Also, the y-axis is again, it's the pure corrected expression?
It's not pure correct, it's the actual expression. And I think, I'm pretty sure, this is Giovadi's LCL data. Yeah.
Actually, I'm still confused. It looks like the y-axis is observed expression, but it's not on the same scale as the x-axis.
Oh?
Right? So the observed expression.
Oh, you're right. We didn't standardize here. Yeah.
But the case of the y-axis is simply not normalized.
Yeah, this may be just, it wasn't normalized. This was the observed data. I can't remember which one exactly, but it wasn't normalized, yeah. Yeah. Yes?
On the previous slide, are you doing 2 rounds of cross-validation? So is it like cross-validation to get the...
We are doing that now, actually. Yes, we didn't do it before, yeah. But it doesn't change hugely. It changes a little bit, yeah. We are being a little bit...
Just choosing lambda is not that much.
It's not that much, but you can see the effect a little bit, yeah. I have some plots for that, too. Okay, so, we can predict gene expression levels, so that's good. So how much time do I have?
Well, not that much.
5 minutes, 10 minutes? No. Yeah, I have plenty. So I'm 2/3 of the way. So summary, we can predict gene expression levels. I can always predict. Accuracy may vary, but so local contribution we can reliably estimate. Distal is harder, so we gave up on that. And the genetic architecture, at least with the sample sizes we have, we only can see the sparse component. So that's why we chose ElasticNet to do this prediction.
Do you have any idea of guidelines of what are the optimal sample sizes, or?
The more, the better.
Sure.
But what's kind of cost included? Yeah, yes. I'm not sure. Yeah, we haven't looked into that. Yeah.
I guess a different way to phrase the question would be: are we approaching the limit of what we can predict for most of these genes? Like if we just get more and more ETL samples. For some of the genes, there's nothing really.
Yeah, ARAP2, you get it right. Yeah.
It seems to me like if you had less data and you had more biological prior in how to combine, like how to put prior weights on the SNPs.
You should do better.
You should actually see
It's a trick here because there's LD there. So if on a local within a gene, let's say, I don't know, within a megabase, or I don't know what's your definition of local there.
Yeah, 1 megabase
There's
on each
strong
side.
LD between all of them. We don't
Yeah,
know
but the function
But, yeah.
break that already. That's the
A little bit,
like,
yeah.
If you look at like at least
Sure.
not coding
But for
regularly,
prediction, it's not that. It's not that. For prediction, it doesn't matter if you predict it with a tag variant or with the true call.
Yeah. But for the application, it does matter, yeah.
For the application, it would
Yeah.
be terrific. Yeah.
Yeah. Okay. So we're going to apply this to study the function of every human gene. Okay, so if we want to know the function of a gene, not human, so of any gene, like typically what people would do is you would pick a model organism, knock out the gene, and see what are the phenotypes of this organism. So that's what the International Mouse Phenotyping Consortium is doing. They generated 20,000 knockout mouse lines, and they have phenotyped many of these. So we are going to use this, but kind of mice are not human that we know of. They're smarter than humans according to this. I was reading the Hitchhiker's Guide to the Galaxy. Anyways. Okay, and we know that we cannot do experiments in humans. So, and then we know these natural human knockouts come to save our day. And so, these are people with loss of function mutations in both copies of their genes, and so they end up being natural experiments. We didn't have to experiment on them. They just came like that. We study their phenotype, and that helps us understand the function of genes. So this is not really what I do, but I think it's a way of explaining what I want to do, kind of in an easier way. So I don't know if you have followed this literature, but there have been many news kind of perspective last year in Nature and Science talking about the dawn of the Human Genome Project, and this is very exciting things that's going to happen. This is great. So as I was saying, so to come back to what I would like to do, so the Human Knockout Project is this systematic effort to understand the consequence of disruption of every human gene. So the way I think of it is, if this is the distribution of gene expression level of a given gene, the knockouts will be here, so they are predicted to have zero, or at least the functional part component of it to be zero, and then the rest of the people will be here. So these projects are sequencing tens of thousands of individuals, and then you identify a handful of knockouts, and those are very useful model systems that we are going to try to learn a lot from them. But we have all these remaining people where they will have some variation because of their genetic profile, and we should look into that. So my goal is to learn about the function of genes just looking at this variation of their expression levels. So for that, we're going to need transcriptome data and phenotype data. So this is what we need. We need transcriptome and the phenome. And for that, we have, there are all these biobanks that are being collected, where with UK Biobank, we have 500,000 people, more than 3,000 phenotypes. Depending on how you count, we could say infinitely, no, not infinite. But anyways, we have the Precision Medicine Initiative, so a bunch of biobanks, 1 million individual level kind of biobanks where we have their phenomes and we have their genotype data. So we have their genomes and their phenome. So we still need the transcriptome. So since that's the reason why I talked about the prediction of gene expression level, so given the genome, we'll try to predict the transcriptome. So that's what we do. So now we have the transcriptome and the phenome, and the idea is to learn about the biology, the function of the genes by looking at running these associations types. So then we found out that according to the Broad, we have 500 million people genotyped. So in order to use this, we need to be able to take advantage of this. And so in this case, getting access to all this data is almost impossible because of computational storage problems is one, but also kind of regulatory, like consent issues, and also investigators not really willing to share their data. and things of that sort. So we want methods that can take advantage of the summary results of these GWAS. So we need to leverage these GWAS results. And so now let me go back and repeat what I said, but in a slightly different format. So what we propose to do is to take the genotype of individuals and the phenotype, and the genotype here is the genotype matrix where rows are individuals and columns are genetic markers, or SNPs for simplicity. And what we do is we take this genotype matrix, we use the prediction models that we trained using the GTEx data and other data sets where we have RNA-seq data and genotype data. And so we generate this predicted transcriptome, and then with the predicted transcriptome, we run the association between the phenotype Y and each one of these columns in our predicted transcriptome. This is the method that we call predict scan because we predict expression and then we scan the genome, but now it's more widely known as transcriptome-wide association study. But predict scan kind of gives you the idea that it does not have to be just gene expression, it could be other molecular phenotypes as well. So to put it in context, a GWAS study, what you would do is you would take your Y, your phenotype, and run linear regression on each one of these columns. So that's represented here. And what you end up with is this SNP level result. So this is your Plink output, or whichever your favorite program to run GWAS. Whereas PredictScan will give you gene level results. So what we would like to do is to go from the SNP level result to the gene level result. And so we work out the algebra so that we can do that. So the reason why you can do this is because essentially a GWAS is if you normalize the phenotype and the genotype, the effect sizes are just the correlations, right? The same is here. So this is correlation, and no, here. So here we do a linear combination, which is our predictions are linear. That's why we like to be linear. So we do a linear combination of genotypes, and then we run the correlation with the phenotype. So that's, whereas here we calculate the correlation, and then we do linear combination. So because things are linear, we can go either way, and that's why we can take advantage of summary results as opposed to needing the individual level data that can be very hard. So this, I think, is the only way we can really scale up to billions of samples. But please let me know if there are other ways in which we can be nonlinear and still be able to scale up. Here is the formula. I don't know how much. Okay. So yeah, so this is the Z-score of these associations, which with the Z-score you can calculate p-values. You have the direction of the effect. So the Z-score is a function of the Z-scores of the GWAS results, and then the weights that we trained in our when we did the prediction model training. And here we have the standard deviation of the SNP, and this is the standard deviation of the predicted expression. So these are things that we would need the individual level data to calculate, but instead of using the individual level data, we use reference data to do that. We can use 1,000 genomes, or we can use, actually we ended up now using the training set, which we found to be, the prediction model training set we found to be more robust. And here is how well it does when the reference set where you calculate the LD is misspecified. So here is a study that was with African individuals, and then the reference that was used to calculate the variance of the genes, so which is essentially the LD of the SNPs in the prediction models. So if you use the right population, you get pretty good correlation between the approximate summary-based method versus the individual method. That's the not approximate one, the exact one. But if you change so if you use East Asian study so your GWAS individuals are East Asians and your reference sample is African, the performance gets, the correlation, the approximation is not as good, but still good enough to find the associations. So this is the pipeline that we implemented. We took GTEx, 49 tissues from GTEx. We trained our prediction models. We put them into our calculator of the association between the genes and phenotypes using about 3,000 GWAS and meta-analysis GWAS results. We go through some filtering steps to avoid early contamination that I won't go into. And then we save them into a database that we are calling Gene2Pheno. So this is accumulating all this information about association between genes and phenotypes. Here is a screenshot of the database that you can check. We have also made web applications for easy implementation. And I think I will just skip to the conclusions. In summary, so I went through some prediction methods, all of them based on linear additive genetic models. So we used these models so we did the prediction, and then we applied them so that we can, in order to learn the function of genes. So here are my collaborators, and thank you.